Hi, Christina. Thanks for sharing with us today. If you would just start by telling us a little bit about yourself and the research specialty of your lab and a little bit about what your role is within the Mucosal Immunology and Biology Research Center.
Sure. Thank you for having me today. My name is Christina Faherty, and I'm an assistant professor in the Mucosal Immunology and Biology Research Center at MGH. And I primarily focus my research on bacterial pathogenesis, specifically bacteria that infect the gastrointestinal tract and cause diarrhea.
So would you tell us a little more about which GI pathogens specifically you study and kind of why you are interested in studying those specific pathogens?
Sure. I mostly focus on a pathogen called Shigella, but I also do some other work with pathogenic Escherichia coli or Salmonella, which you may have heard about. Basically, and especially for Shigella, it's a pretty significant global pathogen. It causes millions of infections each year. A lot of these infections occur in the developing world and in malnourished children. And also what's really interests me about these pathogens is that they really have a lot of hurdles to overcome when they cause infection. Just by transiting through the gastrointestinal tract, they have to overcome things like acid in the stomach, harsh conditions in the small intestine, limited nutrient and competition in the large intestine. And so it's really fascinating to me that these pathogens are able to overcome all of these challenges and still establish successful infection. Despite over 50 years of research, vaccine development has been very difficult and has routinely actually failed, unfortunately. So I'm interested in trying to find new solutions to help facilitate vaccine development. For some reason, for Shigella vaccines and even other enteric pathogens, despite knowing a lot of the mechanisms of infection and how the bacteria cause infection and even how our immune systems respond to the bacteria, we still haven't been able to replicate that enough successfully to create a successful vaccine. So one example is with Shigella is we know some antibody responses that are developed in patients that have become infected with Shigella. And this does provide limited protection against reinfection if they were to be exposed to Shigella again. However, anytime we've gone into the lab and tried to replicate those antibody responses to develop a vaccine candidate and then have tested them in humans, these candidates have routinely failed. So I feel like there's a lot of information that is still unavailable and things that we still don't know about that will help us understand infection better, that will allow us to figure out how we can develop a successful vaccine and how Shigella is so good, even at times, sort of subverting our immune response and trying to fight against it.
Could you walk us through your current research projects and if there's any exciting research findings that you want to share with us?
Sure. So since Shigella is a human-specific pathogen, it's really evolved with humans over the course of evolution. It only really infects humans and some higher primates. So it really doesn't infect other types of animals. So we try to replicate in the laboratory setting as best as possible these human-specific conditions, because we feel like if we can replicate the environment that the bacteria is in immediately prior to infection, we'll be able to identify a new understanding of how infections caused, and then maybe even identify some novel vaccine targets that aren't really expressed in laboratory media. So one thing I really focus on is that the bacteria need to transit through the small intestine in order to reach the colon for infection. And Shigella spends a good majority of its time prior to infection in the small intestine. So when they're there in that environment, the bacteria is exposed to bile. And bile is supposed to help you digest food and break down food products. And it even helps you to kill any potential harmful bacteria. But a lot of these enteric pathogens, including Shigella, are able to resist bile. And so we've gone on and started to look at how the bacteria is resisting bile. And also we found that the bacteria has utilized bile as sort of like a host localization signal, where it almost senses that it's in the small intestine and begins to alter gene expression profiles so it can actually prepare itself for infection in the colon.
So when you're doing all of this research, could you tell us what model types that you utilize and walk us through how you use organoids in your research?
Sure. So for some human-specific conditions, we're able to add a combination of bile salts and glucose to laboratory media to sort of mimic that small intestinal environment. So in those conditions, we've been able to really document how those resistance mechanisms happen. We've identified that Shigella uses these things called efflux pumps that can actually remove the toxic bile compounds out of the bacterial cell. And then we've also identified that the bacteria forms this little protective niche called a biofilm that helps them transit through the small intestine. And what is actually really interesting, since bile is mostly localized to your small intestine, and you lose a majority of it as you transition to the colon, we can actually replicate that in the lab setting. And so we can remove the bile signal and find that they disperse the biofilm and they've entered into this really hyper-infectious state where they're ready for infection in the colon. So then another aspect that we take in with our research to then look at the virulence end of things and how they're causing infection once they're in this state and then can infect colon cells is that we use an intestinal organoid model developed at the center by Dr. Stefania Sanger. And this model is actually really replicates the human gastrointestinal cells that are present, the multicellular types that are there. It's a non-cancerous cell line. So it's really more physiologically relevant. And we've been able to show once we mimic this small intestinal bile transit exposure environment and scenario that once they get into the colon, they can actually adhere better to the epithelial cells and then invade better. And the adherence is actually an interesting story because the bacteria express these proteins called adherence factors that allow them to attach onto the cells. And it was actually a surprising finding because it was believed that Shigella doesn't express these types of proteins. And that's true. When you grow them in laboratory media, you don't see these adherence factors being expressed. But then once you grow them in media with bile salts and glucose, we can see the adherence factors. And then, so now we can have a better understanding of those early steps in infection and now possibly lead to better targets for vaccine development purposes.
Well, thank you so much for that answer. And your talk about the future holding treatments, hopefully that will lead to vaccines. Can you tell us a little bit more about some novel therapeutic options and your collaboration with MIT?
Yeah, so as for vaccine development purposes, as I've mentioned, we hopefully will have these nice new targets to go after. But then we can also maybe look at some other therapeutics that could be really interesting, like novel compounds that might inhibit, say, the efflux pump function of the bacteria so that they're sensitive to antibiotics or something like that. And then we've also been working with Dr. Timothy Liu, our collaborator at MIT, and we're working to help develop bacteriophages, which are viruses that specifically target bacteria, and use that as a potential therapeutic against Shigella. And in that setting, we've been able to show that the bacteriophages that we're trying to direct specifically for Shigella is that they can target the bacteria even after they're exposed to these human-like conditions, and that they are really effective at helping to limit infection of the colonic epithelium, and we can do those analyses with the organoids. So we're hoping some of this stuff can really move forward and give us some new pathways to pursue, especially when you have to consider things like antibiotic resistance, in addition to the failed vaccine candidates that have occurred throughout the years for Shigella. I would like to thank my laboratory personnel and collaborators, both past and present. We've come really far in our research and we have a nice long way to go, but we've definitely built a strong foundation to keep moving forward, and we're definitely excited to continue our research, especially here at the center.